Hello there. Thank you for joining me on this lecture video again. So, on this video, we will be looking at some chemical aspects of hormones in a little more detail. How about let's get started? So, hormones can be divided into two main classes. Firstly, amino acid based ones. These are, for example, peptides and proteins. And the second group is steroids. These are all made of cholesterol. Remember from our nutritional talks in BIO201 and BIO156, some amount of cholesterol is needed in body, but there is no need to get it from food. Your body makes all that is needed. And finally, I want to mention that this classification may very well get expanded in the future. Some scientists are considering eicosanoids as a hormone, while others still classify it as a paracrine. What will happen in the future, we will need to see. But notice how the fields of science are constantly changing and concepts are being re-evaluated and revisited. This is what I find so exciting about working in this field. We are far from a finished product of full understanding these concepts. Okay. Let's look at the actions of hormones in a little more detail. So, have you ever wondered about this? Since hormones are passed through the bloodstream to everywhere in the body, how come we do not see these hormones' effects and changes that they cause everywhere in the body? Yeah, well, you might not have thought about it, but I certainly have. It is because only the cells with receptors for that hormone will be affected. These cells that have receptors for a particular hormone are known as target cells for that hormone. And what they do in the target cell once they bind to the receptor is that they affect the cell's activity in one way or another. So there must be a specific fit or match, if you wish, between the hormone and its receptor. Only the ones that are an exact match will do. One way to think about this is like they would be a key and lock pair. There are many hormones, just like there are many keys, and there are many different receptors, just like there are many different kinds of locks. But only a pair that is a match will bind correctly and alter the target cell's activity. So, what do I mean by when I say that the hormones affect the activity of a target cell? Well, here I have a list of possible hormone activities. As you can see, there are many. They can change what gets into the cell and what is blocked out, or they may trigger or stop production of enzymes or proteins. They may also cause the cell to secrete something, or they may even trigger cell division. So there are many possible effects on the target cells, and these are only some examples that I can think of. Another thing that I want to highlight is that the chemistry really matters here. How hormones act depend on their chemical nature and receptor location. And there are two main ways. Let's have a look at those. Just like you remember from our earlier discussion of vitamins in your earlier biology classes, there are both water and lipid-soluble hormones. Let's start with water-soluble ones. These are typically amino acid based and act on the surface of the cell on its membrane. So therefore, as the receptor is located on the cell membrane, additional extra steps are needed for them to do their job. And these multi-step processes need second messenger in the cell. Your textbook goes into a great detail of this, but I think that we will just leave it here for now. Lipid-soluble hormones instead are typically steroids and thyroid hormones. Because they are able to enter into the cell, they act on a receptor that is located inside the cell. and their actions are much more direct, acting on DNA and through that controlling protein production. So there is no need for these multiple extra steps like the water-soluble hormones had. So that's a rather nice comparison of these two, isn't it? I also want to talk a little about the release of hormones. This is a good time for us to remind ourselves about one of the very basic concepts that we have talked about in our previous courses. I told you that it would be a fundamental one, and we would come back to it often. And this was, of course, homeostasis. So homeostasis referred to the maintenance of constant internal conditions. And we had this rather nice simple diagram that I proposed to you. Remember, something changed. It was noticed by a sensory organ or a sensory structure. Message relayed typically to our brain, where we had a range of acceptable conditions. And if the stimulus fell outside this acceptable set point that we had established, something needed to be done. And this something was done by an effector. And typically this was to stop something from happening. And this was what we knew as negative feedback. I have included a link to the video where we first discussed about homeostasis in our earlier course, in case you have forgotten this concept. Remember that negative feedback was more common. We stopped the change from happening. You could say that we stopped the deviation from the set point. However, there was also positive feedback, which is much more rare, but can also happen. It takes the deviation from set point even further. So, going back to the release of hormones. Levels of hormone in the blood are controlled by this negative feedback. And they are maintained within a rather narrow window of set point of what is an acceptable amount of hormone in the blood. If it deviates too far from that, the negative feedback control loop kicks in. There are two ways the hormone release can be triggered. Either by endocrine gland stimuli, which is hormone release being caused by an alteration in the level of iron or nutrients, for example. Or by a nervous system. This is nerve fibers stimulating the hormone release. Or it can be that hormone release is caused by another hormone. This would be known as hormonal stimulus. I just didn't list it here separately. Finally, let's look at the target cells a little more. These are very specific. Target cells must have specific receptors to which the hormone binds to. Remember our key and lock mechanism, where they needed to be a match. So what affects this? Well, I have three different points to highlight here. The amount of hormone in the blood, amount of receptors in the target cell, and how well the hormone and receptor bind together. The amount of hormone around will also have an effect on the number of receptors in the target cell. If there are low levels of hormone around, the target cell makes more receptors. This is known as upregulation. And if there are high hormone levels around, the target cells will lose receptors. This in turn is known as downregulation. I guess that what I want to highlight here is that the most important take-home message is that the human body is very adaptable. And it is the changes that we especially note, such as, in this case, the changes in the amount of hormone around. The last two big questions that I guess that we should talk about here are, how do these hormones travel in the body? And what removes these hormones so that the levels do not just keep building higher and higher? Let's have a look at these two questions. Hormones circulate in the blood in two possible forms, either as free or bound to the carriers or plasma. And this is why when we do blood work on the patient, we get very direct information about the concentration of this particular hormone in the blood. And the concentration can be explained by two factors: how much or fast the hormone is released, and how much or fast the hormone is inactivated or removed. So I promise to look at also what removes hormone from the blood. Let's consider it. So hormones can be removed from blood by degrading enzymes, kidneys, or liver. Remember, both kidneys and liver have a main task of filtering the blood. And this gives us a concept of half-life. Half-life of a hormone is the time that it takes for hormone concentration to decrease by half. And for some hormones that may be just minutes, while other hormones, it can be weeks. So there is a lot of variation here from one hormone to another. And this depends on whether the hormone in question is water or lipid soluble. This is a rather nice table that I put together based on your textbook summarizing the differences between water and lipid-soluble hormones. You can study it in more detail on your own time. Finally, multiple hormones can be acting on the same target cell at the same time. So let's look at what kind of an effect that would have. This can either be due to the hormone in question needing another hormone to be present so that it can have an effect, or if another hormone is present, it may amplify the effect of the hormone that we are interested in. Or this other hormone may act against of the hormone of our interest. So that's it. On this video, we have covered a lot. We have discussed the chemistry, actions, release, target cell specificity, and finally transport and removal of hormones. I know that it has been a little theoretical, but as I said, over the rest of the course, we will refer back to the different aspects of endocrine system on multiple occasions as they relate to the very system that we are looking at that time. I will see you in last of our lecture videos for this chapter.